Hi, my name is Paul Marrone. I'm a teacher at James Caldwell High School. I teach science. I teach two subjects. I teach Biology 1 for 9th and 10th graders. I also teach a research and molecular genetics class for juniors and seniors. I've been teaching for the past 7 years, and I really enjoy it. I'm really enthusiastic about it. My background is mostly in molecular biology. As for the molecular research and molecular genetics class, that's my field of study. The Toshiba America Foundation grant we received is actually for that class. That class is a genetics class that's also affiliated with Rutgers University's Waxman program. And the Waxman program is a program where schools across New Jersey, about 20 schools, 21 including mine, where we use molecular biology techniques and tools to sequence and analyze the genome of various organisms. What this means is, for instance, for the past 3 years, we've sequenced the genome of brine shrimp, Artemia franciscana. Hasn't been sequenced before. The students took the entire project from beginning to end. They have a library, and then they sequenced and manipulated the genome and analyzed it and presented their findings. The organism this year that we're doing is called duckweed. Duckweed is an important organism. It's the smallest flowering plant known right now to man, and it grows in fresh water. You've probably seen it across ponds. It's an important food source, a very cheap, sustainable food source. It also is useful for eliminating contaminants in fresh water, so it can be used for sequestering any kind of contaminants. So we're sequencing this organism through the Waxman program. When I first received my Toshiba grant, I received 2 of them actually. It was when I first started teaching this course, and we didn't have any tools to do any of our research with. So even though we're a part of the Waxman program, there were no tools for us to use. No micropipettes, no gel chambers for doing gel electrophoresis, no thermal cycler, no hot water bath, no vortex. So with this grant, actually the 2 grants I received for 2 years, we used the money to purchase the equipment we needed through Bio-Rad. And the equipment that we're using for our project now is still the same equipment that we purchased years ago through the Toshiba America Foundation grants. What I'm going to have is the students lead you through the different parts of the project that we're still doing. Even though the grant was from a few years ago, we're still following the same steps through the program, and I'll have them guide you through the different steps of the program. The importance behind this particular grant is that without it, we wouldn't have been able to do any of this research. The class wouldn't exist. The tools are actually being used in addition to the Waxman program's project. We continue to use the tools throughout the year for other things too. So besides sequencing the genome of an organism, we do other things like DNA fingerprinting, Western blot, SDS page, and without the equipment, the students wouldn't learn nearly as much as they could by doing the hands-on lab work. So there are schools that are part of the program at Waxman that don't do as much as we do because they don't have the tools. And they may have some loaner tools from the Waxman program, but not enough for their classes. So the students in those programs aren't learning nearly as much as the students, say, in my class, who actually get to do the project from beginning to end, and there's enough equipment for everyone to use. So I'm going to take it over to the students right now, and they'll lead you through the different steps of the experiment and explain what they're doing.
This is Mr. Marrone's research and molecular genetics class.
What you need to do now is dilute your cultures.
Okay.
Because we need a dilute sample to do PCR. So you're going to dilute your samples like this. Take 5 microliters of your culture and add it to 200 microliters of water. Make sure you correspondingly label these.
Okay.
So you're going to make 1 for each dilution.
Right.
And then what you're going to do is take the dilution and add that. It looks like you're going to add 2 microliters of the dilution and 23 microliters of your master mix and add it to the right corresponding PCR tube.
I am taking some sterile water, 200 microliters of it, and I'm pipetting it into the microcentrifuge tubes so I can dilute our DNA.
And I'm taking 5 microliters of our DNA and putting it into the microcentrifuge tubes and mixing it with the water so it's diluted.
We're preparing our bacterial cultures for PCR so that we can tell if their inserts are big enough to be sequenced.
And we're doing that by diluting our overnight cultures which we inoculated yesterday with water.
Alright, So we are putting in our ladder into our first well here so that we can use it to identify how many base pairs we have in each sample of DNA that we run.
Talking about the loading die?
And what do the red and black strips mean on this?
The red and black strips differentiate the two different gel processes that we're running. You can run, I believe there's 8 on each, so there's 16 altogether, and you just run it for a shorter amount of time and you get more information out of it when you're done. Alright, so what we see here is we have two hooks here onto which we're going to plug an electrical source so that we can run a current through the buffer and through the gels, because DNA is negatively charged and will move from negative to positive along with the current. Alright, so now that we've run our gel, I'm going to remove it from here, and we're going to go bring it over to the UV light so we can see everything that has happened in the gel. And we're going to turn it on. Okay, there you go. And now we're going to take a picture of it.
And what is this we're looking at?
This is what we are looking at is, here you have the ladders on the side, and then within here is each individual PCR trial, and it shows how large each insert is that ran through. The longer ones stayed over towards this side, while the shorter ones ran through further.
This is our PowerPoint analysis of 2 of our clones.
It helps if you go like this.
In this first lane is the DNA ladder, and then in this one is the uncut, which means the plasmid DNA wasn't digested by restriction enzyme. And then here is the cut, and that's the sample plasmid, still the same clone, that was digested by restriction enzyme.